Hello, I'm Zach, and here's my thesis project. We're calling it Phylo-TSS, and we're kind of looking at kind of using machine learning models to predict transcription start sites within plant genomes across different groups of species, seeing how well models trained in one species do in other species and what insights we can draw from that. So just a quick introduction to that. So we're interested in looking at promoter architecture in plants. And so we have proximal promoters that are up close to the transcription start site, usually within about 2,000 base pairs of the transcription start site. And those kind of regulate gene expression. Right, and so changing the promoter region can alter when, where, and how much of a gene is transcribed. And if you attach the right promoter to a gene, you can actually cause it to be transcribed in the way that you want it to be, when you want it to be. So I've got a little schematic here. So you've got our little purple RNA polymerase, and an organism wants it to bind to DNA. So we've got these binding signals in the DNA. They're just a sequence of nucleotides that are recognized. So the RNA polymerase will bind there, except on its own, that's usually not enough. And so the polymerase will usually just float by and won't really stick around long enough to bind and actually start the transcription process. But there are transcription factors that occur in the promoter region, and those can assist in binding the RNA polymerase long enough to get it to initiate transcription. So that'll stick on now if you have the right combination of transcription factors in the right conditions. And then the polymerase can move on and start producing a nascent RNA as the first step in the gene expression process. The problem is promoter regions are rather big, and you have a whole menagerie of different transcription factors that can occur there. And you don't necessarily know, just looking at the sequence, which ones are going to occur there. They can vary a lot in how many are there, which ones you have, and where they are related to each other. And they don't even necessarily have to be up next to the transcription start site. You can have RNA, you can have transcription factors that are rather far away, but because the way that DNA is flexible, they can bend over towards it and still play a role in this process. So it makes it rather difficult, or at least a lot less intuitive, using the sequence of the promoter region to predict transcription start sites. In addition to that, the actual sites where these transcription factors bind are variable as well. So they have sequence preferences where certain sites are much preferred for the binding of the transcription factors. And so you can see on this example diagram, we've got, I think there's okay, I won't bother looking for the pointer tool. But you can see at different positions, we have different heights in the schematic, and this shows the preference that a given transcription factor might have for different nucleotides at different positions. And these will vary depending on the transcription factor. And so it just shows the sequence doesn't necessarily need to be highly conserved for a transcription factor to bind equally well at various different sites. So when we talk about promoters being similar, sequence conservation is not always such a good metric to look at because the conservation can be rather weak, especially over more distantly related organisms, just because the order in which and the yeah, so certain TF binding elements might be absent or present in different organisms or in different quantities in different places. And even those, even when the same ones are there, they can vary in their own sequence. But some things are conserved. So some TFBSs, the transcription factor binding sites, are a lot more heavily weighted in how the organism considers I shouldn't try to give it agency, but they're a lot more essential for the transcription process. So they tend to be conserved, and their relative locations might be important. And so it'll be interesting to see how this promoter architecture, the relationship between the different TFBSs within a promoter varies between species, especially because we can use this to try to generalize assumptions about promoter architecture from a model organism, say, to a useful like agricultural crop or a medicinal plant. So it'll be kind of the incentive for this project. So we've got a few different organisms that we're interested in looking at. We've got rice, oryza, corn, zea, in the Poaceae family. So they're kind of our close group. And we've got Arabidopsis and Catharanthus, which is a plant grown for some medicinal products that it produces, which are rather distant from each other and the other two. So we'll be using a machine learning model. This is useful because we can take lots of data for different genes where we know approximately the promoter region and we know the transcription start site location. And we can feed it lots of data, which wouldn't be feasible to try to give to a human, and wouldn't be feasible to try to construct some really simple like math equation. But we can use a machine learning model, which will kind of produce a mathematical model for an optimization process. And then we can use, for example, the parameters from the model to draw insight about the TFBSs. About the promoter architecture, and also see how well they compare to each other in different organisms by using a model that's trained in one organism on another. So we'll be using features, which are the transcription factors and possibly their locations within the promoter as inputs, and we'll feed these into a machine learning model. And this produces essentially an equation that has various weights. So we have, in our example, or in our case, we'll be using a linear regression model. This has our weights on the right. So we've got all these different weights that are multiplied by our confidence that a given transcription factor is present. And then it's just fed into a function that helps to kind of break up the classification into two separate groups better than like a linear model. So we have essentially this produces a confidence that our that are TSSs in a given location, given all these data here. So how that works is it starts by making a guess about the different weights, and then calculates a cost function, which is basically just basically just a quantification of how wrong it is. So if it gives a really high confidence where it's wrong, that'll generate a really high cost value. And if it gives a really low confidence when it's, if it gives a really low confidence that something is a TSS when it's not, it'll also generate a higher cost value. So then it just calculates the effect of changing those, of changing each of the weights on the cost function and adjusts all the weights in the direction of minimizing the cost function by some small increment, and repeats this cycle until we get an acceptably low cost value. And then our equation is optimized, and we're able to hopefully classify the data, which is at a given position, is this a transcription start site or not? Okay, so all that is for the purpose of taking one machine learning model, applying it to data from one organism, and then getting a function out of it that attempts to calculate the presence of transcription start sites, and then applying that to different organisms to see what insight we can draw from this. So difference in performance can help us gauge the similarity between the promoters, so we can see maybe evolutionary distance is the only thing that determines that, and we'll find a strong correlation. Or maybe there are certain clades that have highly conserved promoter architectures. This is kind of what we're trying to explore. And differences in the weights that the models generate to try to predict these transcription start site locations, and kind of inform us what transcription factors have become more important in different organisms. And then of course we can follow that up with dry lab work. So one thing we can do is we can take our training sets. So this is just the collection of TSS reads, different genes that we know the transcription start site locations of. And then we can feed this into a machine learning model, and this produces the mathematical models. So we could look at the weights, for example, or we could take a given model and test it on a new test set that wasn't the training set, and then another test set from a different organism, and see how well those compare. And so we've done a little bit of work leading up to this. So we've been able to, find, transcription factor binding sites in organisms like C. roseus. And then we're doing an analysis on tissue specificity, which kind of compares to this between the organism study. It's just within an organism, but with different tissues, to see how the models compare. And with that, does anybody have any questions?
All right, Zach, thanks. We're open up for questions. John?
Hi, Zach. Thanks for that. So in terms of thinking about this cross-species, for the transcription factor binding sites, I am assuming you're going to have some database of motifs or PWMs or something like that. Is that correct?
Yes. Well, yeah. Well, I'm not sure exactly we'll do it yet. I think we'll take a collection of PWMs that seem highly likely to be of significance, probably as many as we can get, and just try to minimize that as we build our models, so that we're not producing too many features where it becomes cumbersome and full of a lot of not really interesting features.
Okay. And those motif PWM type models are they primarily built from sort of biochemical experiments from Arabidopsis? So what I'm sort of curious about is, as you go across phylogeny and evolution is happening, then there's the potential for the transcription factor, the protein sequence, to change, maybe slowly,
Right.
but in terms of sort of what the sequence that it's going to bind to, and I don't know whether anyone has done experiments to sort of look at how well conserved particular target binding sites are across plants, for example. And I know people use Arabidopsis data to like look for motifs in maize, for example, but I don't know sort of how how reliable that is. And I'm wondering if you
Yeah,
have any thoughts about sort of if there are data and how you would address that.
right. Yeah, I guess I don't know yet if there are data for other organisms, for example, that I could use to address that. But that definitely is something that needs to be addressed.
Yeah, I mean,
Yeah, I guess.
there's sort of the like your motif showed, there's already this variation. The transcription factors sort of have they have some wobble, right? Some in terms of what the sequence they will actually bind, even within the particular organism.
Right.
And I just don't know how much that changes over evolutionary time. And I don't know whether, that might have been looked at better in, fungi or something, if people haven't looked at it in plants, and maybe somebody else has some thoughts on that.
Yeah. That. Yeah. I'll definitely be addressing that.
Okay, thanks.
Bruce?
So I have a dumber version of the same question, which is how do you choose your features? And I'm saying dumber because John knows way more about what you're talking about than I do. I'm thinking of it more in a machine learning regression format. It seems like there's an extremely large number of possible motifs or features or lengths of motifs even that you could use as features. How do you narrow that down?
Yeah, that is a really good question. So I suppose we could try to narrow it down by having a cutoff for maybe expression levels. If a feature isn't highly expressed, we could, if a transcription factor gene is never highly expressed, we could maybe assume that it's not terribly important. That might be a good place to start to narrow it down a little bit. And we can use L1 regression, which kind of weeds out. We start with a huge data set still, but it kind of weeds out some of the features as it goes along optimizing. But yeah, that's all I can think of at the moment.
Okay.
Sam?
Actually, very related to that, I was wondering if you imagined future versions or extensions to your model that would sort of address the sort of syntax or relationship between elements and how maybe in evolutionary time, as a repressive and a promoting transcription binding site get further apart from each other, they might have different sorts of outputs. And sort of how difficult would it be to extend your model where you have the sort of linear contribution of all the different binding sites to sort of looking at them jointly, or how they might act together.
Yeah, I see what you mean. Yeah, because that's not really addressed so well in a linear model. If you're talking about like how different combinations of TFs can maybe have a different impact. I have thought about that. I don't know how much time we'll have to dedicate that, but I do think it'd be interesting to look into that and maybe try a different, maybe a different machine learning format, like a neural network, and see if that can give more accurate results. But we do see, I think we do see pretty accurate results with linear models already. So I'm not, I don't know if I'm experienced enough yet to say, but I'm not sure how much we can improve it.
Great, thanks.
Any other question for Zach?
Yeah, I've got a question actually. Is there going to be any direction at some point, or will there be sort of the architecture in place to begin incorporating more ancestral plant lineages, and then sort of keep comparing, like, you know, how these sites are differentiating as we, you know, go deeper in the phylogeny?
Yeah, I certainly hope so. It kind of depends on how much data we can get. So we have to sequence TSSs from plants. It's basically just doing RNA-seq, but trying to pull out the transcription start sites. I'm not sure how much data exists on that yet. But yeah, I think once we have the methods in place, we could apply it to any pair of organisms.
Awesome, thank you.
All right. Any other questions for Zach?
So I'll just jump in there for 1 second and say that Zach answered the questions really well. These are some provocative questions where I've left Zach a little bit. We haven't talked about every single thing, but he's answered the questions well. And if folks would like to go deeper on those aspects, we can talk at a future time. Nice job, Zach.
Thanks.
All right, well, let's unmute and give Zach a round of applause, please.